Okay, so for the second part of our lab, we're going to be doing principles of inheritance for blood types. So we're going to do ABO inheritance. Now, the good news is that it's a Punnett square, and you've done Punnett squares, and the rules are the same. There's some bad news there. Some of you are still struggling with Punnett squares. We still had a lot of wrong answers on those quiz questions, on those test questions. So we will kind of start from zero and review some of these topics. We also do have some information to add on. So we do have some specific rules for blood typing involving codominance, specifically how to set up the Punnett square, the annotations that are going into it, and then how do you treat RH factor versus ABO, because it's a little bit different. So we'll just tack on those rules, follow the same patterns for determining blood types, and we'll go from there. Now, it's, again, still following the rules of Mendelian genetics. Your parental gametes have a 50-50 chance of carrying the maternal line versus the paternal line, so that is identical, right? So there's going to be one allele inherited from each parent for ABO, and then there's going to be one allele inherited from each parent for RH factor, which means there's going to be one Punnett square to determine if you're type A, B, AB, or O, and there's going to be a second Punnett square to determine if somebody is positive or negative. So two different squares are going to be needed to determine the entire blood type. This should all be review, but we'll go over a couple of things anyway. An allele is a different form of a gene, so you could have two copies of the same allele. You could have a dominant allele and a recessive allele. You could have two recessive alleles, so on and so forth. Antigens we know are on the surface of red blood cells. Antibodies are found in the plasma. When an antibody matches up to an antigen, it's going to cause agglutination. So we do not want to have the same antibodies in the same blood as the fitting antigen, right? You do not want an anti-A antibody in the same blood as a blood cell with the A-type antigen. That is incompatible. They will agglutinate, which is a kind of clotting. It's a clumping. It's going to cause blockages. It's going to destroy your kidneys. Okay, so again, homozygous dominant means that there are two, homo meaning same as, two dominant alleles. And then homozygous recessive means having the same two recessive alleles. And then, of course, heterozygous means two different alleles, one recessive, one dominant, and there's just two. So there's no, you don't get to say dominant, recessive, anything like that. You just say heterozygous and you're done. Now, one of the first things that we can talk about in blood typing is the annotation. So we're not going to write A and B at the top of our Punnett square. We're going to write this annotation right here with a superscript A, superscript B, and O is going to be a lowercase I. Why did we do that? My suspicion is it's hard to tell the difference between an uppercase O and a lowercase o. So we just went ahead and did a lowercase I. All right, so let's establish some rules here. A is dominant, B is dominant. Therefore, if you have AB blood, they are co-dominant. So that is an option in the extension of Mendelian genetics. We can have something called co-dominance. Not one erases the other. They express equally and are dominant to other forms. O is recessive. The absence of this allele or the absence of instructions for an antigen is going to be recessive. So if you inherit any copy that codes for these types of surface antigens, then that will be expressed. We did that. Positive is dominant, negative is recessive. So if there's one copy of the D type, the genes that code for the D type antigen, then that person will have positive blood. And again, ABO alleles are separate from RH factor. They're inherited separately. This is going to be one Punnett square. This is going to be a second Punnett square. Again, here's our annotations. I superscript A, I superscript B, both capital letters. Type O blood, lowercase I only. Now, here's another complicating factor. Let's say you know that one parent is type A and the other parent is type B. That person who is type A, that could be homozygous dominant A, or it could be heterozygous A. Either of those end up being the phenotype A. So in order to determine what goes on the Punnett square that when this is the parent, we have to determine what the alleles of their parent. We have to go up an entire generation in order to figure out if they're homozygous dominant or heterozygous. Same for B. The only things we know very clearly is if somebody is AB type, we know that one copy is A, the other copy is B. And if they're O type, we know that's homozygous recessive by definition, because if they inherited A or B, they would be type A or B. So we will have some examples of this, how we're going to go up a generation and determine, okay, is this person capital A, or are they capital A, lowercase I? And that's essentially another Punnett square. We can simplify it a little bit. If you're really good at Punnett squares anyway, you'll get a sense of this pretty quickly. If you are still struggling with Punnett squares, I recommend that you start from 0, build the Punnett square of their parents' generation, and work your way down to the next generation. So some cheat sheets. Rh positive blood, capital D. Rh negative blood, lowercase d, dominant, recessive. Again, that positive phenotype, because this is dominant, this is recessive. Same deal. If you are Rh positive, you could be homozygous dominant or you could be heterozygous. If you are Rh negative, by definition, you are homozygous recessive. Hopefully this is making sense. In my opinion, this isn't for memorization. This is for make sense of it. It's going to make your life easier if it makes sense. Now again, the A allele is expressed in the presence of A, as the presence of A antigens. B allele is expressed as B antigens. O, or lowercase I, is how we write that on the square, indicates neither A nor B antigens. So it's an absence of antigens. Likewise, dominant D allele indicates we are Rh positive. Recessive D allele indicates that we are negative. There are no Rh antigens on the surface of that cell. Now this one, we're starting simple because we already know how to fill out this Punnett square. Parent 1 is it's not homozygous, is it? Because this is AB. It's codominant AB. Parent 1 is codominant AB. Parent 2 is homozygous recessive O. So mom's going to be written like this: I superscript A, I superscript B. Dad's going to be written like this: lowercase I, just because it's easier to see and read on a Punnett square than O. So one parent set of alleles gets put on this axis, the other parent's alleles get put on this axis, and we're simply going to cross multiply. We're going to end up with this A here and this I here. Going to end up with this B here and this I here. This A here, this I here. And this B here, this I here. So now if we look at the outcome, the probabilities are that we've got type A here, heterozygous A, heterozygous A, heterozygous B, heterozygous B. So none of their offspring have type O, none of their offspring have type AB, 50% have A, 50% have B, and again, we haven't done our H factor. We do not know positive or negative yet. Now this one is going to level us up just a little bit because we know mom is type B and dad is type A, but we don't know if they're homozygous dominant or if they're heterozygous, either of them. Homozygous dominant B, homozygous dominant A, heterozygous B, heterozygous A. Those are two options. So we have to go up a generation. So if dad's type is A, dad's mom was type A, Dad's dad was type O, and then we go up. That's mom, there's dad. So if dad's dad was type A, whoopsie, that means he was AA, or A recessive I. If dad's mom was type O, that means that it was homozygous recessive II. So in order to get dad's type A, he had to have gotten one copy. It doesn't matter if it was this copy or this copy, or if it was this version or this version. Either way, if he has type A blood, by definition, he got that A type from dad's dad. So that goes there. With dad's mom being type O, either one of these inherited comes over here. It's the same either way, right? So you don't necessarily have to do the full Punnett square if you understand the principles of inheritance thoroughly. If you don't understand the principles of inheritance thoroughly, you might want to play out a few of these options here and see how that's actually going to work. So do, this on one axis and this on the other axis, and then do another one that's this on one axis and this on another axis, and then circle all the ones that lead to dad's phenotype. Get it? Let me know if you don't. Email me. Likewise for mom. Mom's mom was type AB, and mom's dad was type AB, and she is type A. For me, I've got enough experience that I can intuit exactly what happened there. In order for mom to become type B, she had to have inherited the B from both parents. There's no recessive to inherit when they're both type AB. But again, if you're still working that out, then do this Punnett square and then circle all of the ones that could have become mom, right? So now we know that mom by definition is homozygous dominant and dad is heterozygous. So she's homozygous dominant for B and he is heterozygous for A. So we have these now, and we can create the Punnett square for them. To be fair, sometimes we do not know. What if somebody's adopted and they're type A blood, and we don't know that their parents' generation, or maybe their parents are just old school, or from another country that wasn't keeping good records for a while. Maybe it was, a political coup and everything got burned to the ground. So there's a lot of reasons we may not know the parental generation, one step up in the parental generation's blood type. So it is perfectly acceptable to start a Punnett square and have just a question mark on the second allele if you're not sure if they're homozygous or heterozygous. That's a perfectly legitimate thing to do. And it gets you a certain way into your probability. It's not a 100% accurate square, but it gets you a starting point and makes it so you can make some educated guesses. Okay, so again, we've determined that this was homozygous dominant B. We've determined that this was heterozygous A. And then we start doing our sort of cross multiplying. A goes here, B goes here. This A goes here, this B goes here. This B goes here, this I goes here, that's O. This B goes here, this I or O goes here. So probabilities, 50% AB, 50% B, and heterozygous for B. So the possible blood types of their offspring are A, B, and B. And again, we still don't have positive or negative. That goes on a different square. Now, mom is Rh positive, and for some reason we know that she's homozygous dominant for Rh. So again, if we didn't already have that information, if it weren't written down, we could go to her parental generation. But we happen to be given that mom is homozygous dominant for Rh. So she has capital D, capital D. Dad is Rh negative, so he has the little d, little d. Okay. So probabilities that their offspring will be positive. This D goes here, 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 this D goes here. Yep. 100% their offspring are positive. 100% their offspring are heterozygous for Rh factor. So it's not possible for them to have Rh negative offspring. So this was a source back in the day when we discovered blood type testing. Before we had genetic testing, we've had blood type testing for a lot longer. So this could be used to determine paternity. This was our original paternity test, and of course, it's not 100% accurate. It just gives you a statistical probability. So, for example, if let's see, if this were mom and this were dad, but they had an Rh negative child, then I'm sorry, you are not the father, is how that came out. It doesn't work every time though, does it? Because it's not specific enough genetically. It's just a statistical probability. Okay, Rh positive mom and Rh positive dad have a child that is Rh negative. This is the same question as can let's say a mother with Huntington's and a father with Huntington's have offspring without Huntington's. And the answer is yes, if they're both heterozygous, right? So we do that Punnett square for two heterozygous people, and we're always going to end up with a square with the recessive alleles in it. So we've seen that concept before. So I'm not going over it again. Let me know if you want to go over that again. Now your blood type testing instructions are going to be built into the online lab, so I don't necessarily need to spend a ton of time on this, but I will review it briefly. We're doing ABO blood typing by putting anti-sera, or artificial antibodies, into 3 different wells along with a drop of blood into each well. We're going to stir with different toothpicks, because if we stir with the same toothpicks, we will cross-contaminate. Now, when we add an antibody to a well filled with a few drops of blood, if the antigen is present, it will agglutinate. Therefore, an agglutination response is a positive result. It means, yes, you have that. Right? If an antibody is introduced to a well of blood where there is no antigen associated with that antibody, there will be no agglutination, so that's a negative response. So for example, if you add anti-A antibody to type A blood, it will agglutinate. If you add anti-B antibody to type A blood, it will not agglutinate. Therefore, an agglutination is, yes, you have this. This is a more real-world example of what this looks like. When we illustrate it, let me go ahead to how it gets illustrated. It always looks nice and really, really obvious like this. It's not really as accurate to the real world in my experience. I had anti-sera in my last lab, and we would do this by actually poking students' fingers, getting as many drops of blood as we can. It's harder than it sounds. And really the reactions were pretty subtle, the way that they are actually expressed in this set of slides. So this is really pretty accurate in my opinion. So we've got these wells on a clear plate, and we've put it over some text so that you can see that the agglutination really just looks a little blurry. That's it. That's what it looks like in real world. Actually, the only difference between this and the real world in my experience is the anti-sera do have colors, blue and yellow, so that agglutinated sample does get tinted like a little bit darker and a little bit more yellow. This one's clear. So it's still not perfect. This looks like they do make artificial blood type testing kits too, and that's what this looks like. Okay. But this is more accurate to what we actually see in the lab, so it's good to put it over some text to see, okay, this is clear, this is clear, this is blurry. Now, to read this, this is the A well, this is the B well, this is the RH well, always. Again, I've had these little plastic slides. They always have the same orientation. In my lecture, I had it straight across. That's like on a cardstock type of thing. That's also a legitimate form of this test. They're consistent across these different styles, right? So if you're going across, it's always AB RH factor. And then if you're doing the 3 wells like this, it's always AB RH. The only thing agglutinating here is the B well. There's no agglutination in A. That means this is not type A blood. There's no agglutination on RH. That means this is negative blood. It is B blood. So it's B negative. You know what? I edited this and I didn't need it. All right, I changed it back. So we agglutinated in the A well. We agglutinated in the RH factor well. We did not agglutinate in the B well. That makes this A positive. In this one, I'm seeing agglutination in all 3. So that's a little blurry there, a little blurry there, a little blurry there. See, especially right there, you can't see through it at all. Yeah, that's blurry. So this is going to be AB positive. In this well, I don't see, this is perfectly clear, this is perfectly clear, this is perfectly clear. So we don't have A, we don't have B, we don't have positive, therefore we have O negative. So again, if this one looks fishy to you and you're not really sure, just do a compare and contrast to that one. So this one, definitely, we have all 3 agglutinated. Compared to this one, we definitely don't have any agglutinated. So we definitely have A-type antigens, B-type antigens, and the RH factor. Okay, some more examples. This is no longer part of my assignments. If it was part of Blackboard assignments before, maybe one day. Now again, these don't have a control. I like to have a control because it's a scientific experiment. The control should never agglutinate. It should look like that. If you want a little bit more practice, you can go ahead and read these. I'm not going to answer them for you. Now, this is the table that was already in this slide. I added this one to show you that there is in fact a pattern. Now, you can always just carry this kind of thing around in your pocket if you end up in a clinic that does blood transfusions. That having been said, the level you're at right now, I want you to actually understand what's happening. And in order to understand what's happening, that's what this is really useful for. Every time you're doing a donation, every time you're cross-matching between a recipient and a donor, you're considering the antigens on the donor and the antibodies on the recipient. And this is really useful for that, to really driving home that the antibodies are going to be opposite to the antigens. So if you have type A positive blood, then you will have the antigens for A and RH factor. Your antibodies will be the opposite of that. You'll only have the B type, right? We talked about this in lecture, so I don't want to drive it home too hard. I do want to point out that if somebody is negative, we assume that they have the anti-D antibody. Even if there's nothing in their life that suggests that they've been exposed to the D-type antigen before to develop the anti-D antibody, we assume it's there, just for the purpose of just in case. And also, even if it's their first exposure to the D-type antigen, guess what? We don't want to set them up for future hemolytic disease, right? So we're just going to assume that if somebody is negative, they have the anti-RH antibody, the anti-D antibody, and we do our donation planning that way. So this table will really help you with that. I don't think it's worth rote memorizing. Don't ever rote memorize this. There's no reason to. You can use logic, and after you've used logic for long enough, it's something that you can just do. And there is a pattern to this, right? So if you are donating with a negative, you can donate to a lot more people than if you are that same type of positive. Positive can only donate to positive. As you can see here, let's read this down here. The recipient must have at least the antigens possessed by the donor. So if you're going to give to A, then you at least have to have A, right? Or sorry, if you're going to give to a recipient, wait, let me see. I said that weird, and I'm sorry, I said that weird. Recipient must have at least the antigens possessed by the donor, right? So if you have AB, you need at least A, right? I'm still saying that weird. It's okay to have less than, you can't have more than. That's probably the best way to say that. So A negative can give to AB positive because we have the A type antigens, and so do they. And they're negative. It's okay for negative to give to positive because there's just nothing there for the recipient's blood to react to on that blood cell. That's the way to say that. I think I'm going to not muck it up by saying any more than that. So that's the end of this. Let me know if you have questions. There will be a built-in assignment with multiple choice questions for how to set up Punnett squares and blood donor cross-matching, which we already did a bit of on the worksheet.